So we have a message. This is an excitatory terminus from an excitatory nerve cell. So we packaged a bunch of excitatory neurotransmitter into it. We have a presynaptic inhibitory circuit and a postsynaptic inhibitory circuit. And then, of course, they're all acting upon the same dendrite. This is the receiver. There's only one receiver in this case. All right, this is where you're hoping to pass on some kind of message, excitatory or inhibitory. So, action potential is going down. We assume that this cell becomes excited. We have sodium depolarizing the membrane along the axon through voltage-gated sodium channels. So, letter A is a voltage-gated sodium channel. Shown here, and of course we have the relay between the brakes and the myelin. So those brakes in the myelin, of course, your roads of Ranvier. And of course you can't have a brake in the myelin without myelin sheaths. So C is your myelin, covered up, maintained by, of course, myelinating cells. Now once we get down here, of course, same thing, voltage-dependent sodium channels causing depolarization of the membrane, and then you can't have constant depolarization, you have almost immediate repolarization. So structure E, I assume, is a calcium efflux channel, a voltage-gated potassium efflux channel. Eventually, when you get to the terminus, you have a voltage-gated calcium channel, allowing the influx of calcium. And the calcium acts as the signal for the synaptic vesicle containing the excitatory neurotransmitter, marked in G here, that's glutamate. And H is pointing to the neurotransmitter itself, that's glutamate. So the calcium creates actually a more complicated process, but just know that the vesicle fuses with the membrane and releases the neurotransmitter into the synaptic cleft. All right, this is all review again. So there's a number of different things that can happen. Of course, it can bind to a receptor or it can be cleared. I'll go alphabetically. We may immediately metabolize the glutamate or the acetylcholine or the serotonin or any of the neurotransmitters that we're talking about. In this case, it's a amine transferase. It chews up the glutamate, makes it not work anymore. All right, so that's marked in I. It's just a metabolizing protein. We may reuptake a neurotransmitter. This doesn't necessarily work as well for glutamate because glutamate doesn't really work like this, but serotonin does, norepinephrine does. You release serotonin or norepinephrine, oftentimes it gets taken back up into the presynaptic terminal very, very quickly. In fact, it's a target for a lot of drugs. If you've ever heard of a selective serotonin reuptake inhibitor or serotonin norepinephrine reuptake inhibitor, it's that structure right there that you're targeting. It keeps the neurotransmitter in the synaptic cleft so that it can maintain an effect of some kind. You can also have support cells. These are called, well, they're marked as glia. They're just sort of like supporting cells. They're not neurons. They're astrocytes, or they're just dendritic cells that surround and support the neurons. So they may possess uptake channels. They may have a channel that can take up glutamate. All right, so we're taking up here and here. It's marked with the letter K. Okay. All right, and that just clears the glutamate. Now remember, those glial cells are not neurons. So it's not like they can repackage it and send it back into a vesicle. They just take it up and they get rid of it. They're almost like a white blood cell in that regard. Okay? So finally, we're going to get to what effect does the glutamate have? We already talked about it. Combined to a ionotropic sodium channel. That's your AMPA. Causes depolarization of the membrane. EPSP. All right? We depolarize the cell membrane. Glutamate can also bind to the NMDA. It's ionotropic, but a calcium channel. binding to the glutamate dislodges the magnesium, calcium rushes in, further depolarizing the cell, but more importantly, letter N, carrying on various signal transduction mechanisms through the binding of calmodulin. Depending upon the neuron, it's going to dictate what it does. It may modify the gene expression or any number of different things. All right, and to the right here, we have a GPCR. It's a 7-transmembrane marked region. I don't know if letter O in your legend is marked as GPCR or the internalization and tolerance mechanism or desensitization mechanism, but that's what's being suggested here. If you have a lot of glutamate binding this, what can happen is the receptor will actually be enveloped into the cell. It'll get rid of it to make the cell less sensitive. And that's important because you don't want your cell constantly firing a glutamatergic signal. So that's really what's being suggested here. You're actually removing receptors if it's overstimulated. That's also why if you have, say, a drug user or habitual drug user, or just somebody who's receiving benefit from a drug, they develop a tolerance. They develop a tolerance because oftentimes their receptors are internalized or they're covalently modified so that they no longer bind the ligand. So that's why people have to keep upping their dose. All right, that takes us to O. Before I move to P, I'll just mention Q. Q is also a GPCR, but you'll notice that its effect is very different. Q is an autoreceptor, responsive to glutamate, as marked with the green square. And what's happening is it's a G subclass I, whereas this was facilitating the NMDA. This is causing potassium efflux. Is that EPSP or IPSP?
IPSP.
IPSP. So it's making this cell less excitable. It's calming it down because what the cell is really saying is, you've done your job, you've released your glutamate. Now shut up. That's really what that's doing. It's negative feedback. It's an excellent example of a very tiny negative feedback loop. Right? So that's what Q is pointing to. It's not to be confused with the other GPCRs that are related to calcium efflux. All right? So P, and this is a P. It took me a second, but when it's blown up larger, it's easier to see. P is pointing to the triangles, the blue triangles. That's gamma aminobutyric acid. That's going to be released by inhibitory neuronal circuits. This is presynaptic, this is postsynaptic. They're releasing the same stuff. All right? So P is binding to an ionotropic GABAergic receptor, causing an influx of chloride. Calms everything down. So it's a IPSP. That's right. All right? Same thing. Message comes down to the postsynaptic inhibitory circuit, releases GABA. In this case, it may act on a chloride channel, or it may act on a GPCR. This is a GABA B. GABA A is chloride channel, GABA B is a GPCR, that again, through the function of a GI protein, is going to cause potassium efflux. The net consequence is the same. Hyperpolarization of this cell. Not only that, it can bind to itself. It's a autoregulatory mechanism, so it's actually binding to itself much in the same way that the excitatory circuit did. So it's saying, you did your job, you released your GABA, shut up. All right? And it does that by efflux of potassium. You can also have uptake by these glial cells. You may have uptake in the presynaptic neurons themselves. Those aren't shown here. They're drawn a little bit smaller. All right? So that's gotten us to U. I think that's the last letter. So that's this diagram. If you think of it just as a mass of data points that you're expected to understand, it doesn't work. But if you look at it as a process, and that's really what's being delineated by the ordering of these letters, it makes a lot more sense, and you're able to create almost a narrative of what's happening. You could break it up. You may try to isolate a single synapse and be like, what's happening here? Okay? If it's arranged in a way that doesn't necessarily, isn't as clear for you, you can try to recreate the drawing or find another resource so that it becomes a little bit clearer. Now, stop recording.
All right.